statements, I suppose. Amber, I can't ignore you. I can't ignore you, Claire. You tell me to do it, Claire, but I'm just like, I can't do it. So here is the view. This is, let's look at the tip of it. This is a Monarch butterfly antenna. It is clipped. There's a little bit of injury there. But you can actually see the sensory bristles. The sensory bristles are where the neurons are connecting, which sounds oh my god. Big Amy. See, that's very, very difficult, Big Amy. These commands will get very complicated if we keep adding in all these extra words. We just gotta have it sounds and be simple. Be very simple. Very simple. So Amber, here are. We do need a cheese. Jen, you have your love command, Jen. Timer was a cooldown timer between the sounds command, which is... Oh, I see. Sub sounds terrifies me, Claire. That's very difficult to type out. Alex says, I didn't do anything.
It's time to say something nice about yourself.
Jen, I know you, Jen. You can stop ignoring me? I didn't even start ignoring you, Claire. Jen Stinky Cheese, chat, is forcing me to say something nice about myself, and it is because I love Jen. And Jen, I think, likes me as well. It's pretty awesome. Why are we panicking? It's case sensitive, Claire. Amber, this is the antenna then. Something nice about your stick? Yeah, Jen, I know Jen Stinky Cheese.
Red alert.
I'm panicking. Panicking. Jen, I love your face, ma'am. You got another plushie. NMD, what plushie did you get? Should we look at your plushie that you got? I will pull up your plushie after we're finished looking at the sample. So, Amber, you can see here. Oh. What is the sample once again for this one? This is a antenna of a Monarch butterfly, Claire. So we have the little sensory bristles coming off the side. Yes, Risto, I know you as well, Risto Rock. You amazing human being, you. Risto, why are you so kind and loving and powerful? Risto Rock. Numbers? What do you mean, Claire? I'm not sure what that means, Claire Burr. Please don't make me search for butterfly. It was in box 3, Claire. It was hidden away in box 3. The sensory bristles. Hold on, Claire. Hold on, Claire. I need to... I'm focusing right now on talking about the sample. Ah! Okay, so what are the sensory bristles for? And is it see-through? So it is kind of see-through.
What drink?
Thank you for the hydration, guys. So, it is see-through. It wouldn't be as see-through if it weren't for the preparation. So depending on... No, Theo, you're fine. Depending on how the sample was prepared, there could be more or less see-through. So when you preserve the sample, you put it into a fixative solution, which essentially freezes it in time. It's like formaldehyde. The formaldehyde can have a detergent in it called Triton X. The Triton X actually will form like little pores in it, so to make it a little bit more, it can break down some of the exoskeleton structures and leach out the color. Theo gets it.
Ah, you are wholesome as fat. Yay!
Yes, Claire. We're good. Thank you for the stretch. Thank you, Skinny Skeletor, for the host. So, the sensory bristles.
Dreams, one is not
Good night, Theo!
What does that mean?
Come.
Sleep well, Theo. So the chemicals to destroy just a teeny bit in order to preserve it. No, Theo, you're fine. So, the structure is maintained, Jen. So we're still getting like see this connection point here. Thank you, Grimly. Possibly, maybe, sometimes, not always. This is why also, Grimly, we do those school visits, whether it be virtual or in person. And, you know, sometimes we'll get, like, like, most of the questions we get are very kind and very loving. There were a few, of course, that were, like, can we kill the animal? That's where I'm like, whoa, kiddo, we don't talk like that. But most of them are loosey-goosey. Good night, sleep well, Theo. You can spam whenever you want to, Theo. Theo, that's just, you can do that. It's fine. That's why they're there. The antennae are see-through amber because of the detergent that they were placed in. So Triton X will kind of leach out some of the color. And so it's not actually destroying the sample, but it makes it a little bit more transparent. This could have become even more transparent if we wanted to. There's 2 different solutions that you can put a sample in. One is called, Brady, I'm exhausted. Sleep well, Risto. We love your face, my friend. You are breathtaking, sir. There are 2 solutions.
In dreams, one is not tethered by earthly limitations.
What does that mean?
Come.
Sleep well, Risto. I hope you have a great day tomorrow, my friend. There's 2 solutions. One is called passive clearing, and one is called active clearing. If you put the samples into those, it will become even more clear. There is a certain extent of how clear it can make it, so you can't put in like a crab and like have it become see-through. You can make a crab see-through if you have the shell put into a low level of hydrogen peroxide. You can eat away the exoskeleton without injuring the internal tissues, but it's like a whole like, it's like the formula for different structures and organisms is different, like what concentration of hydrogen peroxide to use, and for how long you leave it in. Because if you leave it in too long, it starts to break down the tissue. Now, active clearing, I can't tell you what's in this solution because it's proprietary. It's owned by a company. You put a sample into active clearing solution, and what ends up happening is, hold on, there's some weird music. Y'all hear that weird music? Yeah. Okay, cool. Something just started playing on YouTube in the background. So active clearing is a proprietary solution. We don't actually know what's in it. It's owned by a company. But what I will say is it's very... You put the sample in an electrical current along with this active clearing solution, and it essentially rips out the fat and protein from the sample. And it works well if the sample is not too thick. Because what you're going to do is if you turn up the electricity too high, stuff gets ripped out of the cells and it destroys the architecture of the cell membrane. Passive clearing is what it sounds like. It's a similar composition, we are told, to the solution, but it makes you just leave it in the solution. You place the sample in and it's very gently removing the protein and making it like... It will make it see-through. I've done it, but I've had to soak samples in the solution for like a month, which is how that's way too long of a time. It'd be brittle. It would become brittle, Big Gamey. So instead of having it be like a solid mass, it becomes like very squishy and very bendy. What you do this for if you don't want to slice the sample. If you want to have, if you want the whole sample to be see-through, so you can image the sample in 3D space. Let me give an example of that. And I know Cliff was asking a question about this sample, but let's look at this here first. Yogurt! Chat, Yogurt, I updated your command. Chat, Yogurt Girl was streaming tonight. Chat, make sure to follow Yogurt Girl. She's on the road to affiliate. Go give that woman a follow. Absolutely breathtaking. A true supermodel, influencer. She's amazing. So, Big Game E, we were looking at a butterfly antenna. Samples? Come on, show me something pretty. And we were talking about clearing tissues. So, why certain tissues are see-through. So, Big Game E, this is a mouse brain, and you can see all the way through it. Let's actually pull it up, make it big, and the way this was actually done. Alright, I guess I'll have to just manually make it bigger. That's fine. Is that they actually cleared the tissue. Whatever you think, Claire. Ask Yogurt Garrow, Claire Bird. We got to make it extra poggers for Yogurt Garrow. So they took this brain, and this one is probably into active clearing solution. I did see Jen said, if I drink it and shock myself, would it clear my fat from my body? So Jen, you are extremely fit and beautiful. Don't you dare say that, ma'am. Don't you dare say that. But yeah, they make it clear a bit, Gamey. So it goes from opaque, like almost white in color, to being totally transparent. And then you can do stains on it. So here, I know you're being silly, man, but it was an excuse for me to call you beautiful, and you, dang it, take it. You take the beauty compliment, Jen. Don't make me go over there. I see you, Jen. So.
Hey, you learn. It's time to see something nice about yourself.
It's fine, Claire. I didn't violate the chat rules. It's fine. It's fine. So this sample actually becomes see-through, and then you have to stain it. So if you want to see particular neurons, so Big Gamey, you wouldn't see these neurons and structures if you just put that brain under the microscope after it's been cleared. You have to mark it with a protein stain. So that is a whole can of worms, which I'm happy. Like, we can get into how you do a protein stain. We're gonna bring up the Wacom because there's no way I can explain this without drawing something out. So, yes, depending on what stage shows, getting close, Big Gamey. So let's say we're gonna go back and make it simple. You guys know how much I love fruit fly brains. It's actually more structurally similar to a mosquito brain, but that's neither here nor there. You guys know how I always show you all the mushroom body in there. That's the learning and memory center of the insect brain. As these little poles, yeah, or like, you know, they're the U-shaped in the ant brain, but they're like the little football fields in the fly brain. So if I put this brain under the microscope, we won't necessarily see the mushroom body, and it definitely won't glow. So what you do is you need to make it glow. So how do you make it glow? We do something called the antibody stain. So what you do is that there are... hold on, Claire. 1 second. I don't want to break this thought. You need to generate an antibody that recognizes the proteins on the outside of a given structure. Whether this be neurons or... it could be a particular cell type. So like... stem cells release a certain kind of protein on their outside. So if like here, let's say that here is your cell. This is just one really big cell, and there's a little protein on the outside of your cell. And among all the other cells in the body... Hi, Jimmy! 1 second, guys. 1 second. I'm in the middle of explaining this. All the cells in your body have a different... like, could have these receptors and proteins that look a little different. So there could be a neighboring cell here. We'll make it a slightly different color, and it has a different things on the outside of it. Right? So you want to mark what this red cell type. That's what you're interested in, Big Gaming. You're curious as to where that cell type is. So you can't mark the cell, but you can get these things here. You can nab those proteins or sugars or whatever on the outside of a cell. Those, you get an antibody that connects to it. So an antibody, like similar to our immune response. Immune responses are driven by your antibodies. They recognize certain molecules on the outside of your cell. Same way like they recognize a bacteria is different from your cells, or a virus is different from your cells because the outside of the cell has a unique barcode, basically. Tell me, guys, if we're still on the same page here. So there's this unique barcode identifier to a cell type of interest, whether it be cells in your body or cells that are invading your body like bacteria or viruses. So in an immune response, your body will have antibodies that bind to this like this interaction happens. So this is the blue is human, let's say, and it binds to the non-native receptor or protein or whatever that's being stuck out on the invading cell. And that's how it's flagged for destruction in the body. But we can use this to mark cells of interest in a stain, an antibody stain. So basically we isolate these antibodies, but we make antibodies against a receptor that's in a cell type that you are interested in. So how do you do that? The quick and dirty way, Big Gaming, chat, you take a bunch of these, we can get these neurons, and we put them into a bunny rabbit. And we're going to give him a little carrot. So you put those cells into a bunny rabbit. Because you're putting fruit fly cells into a bunny rabbit, the bunny rabbit is going to have an immune response against those cells. So if the immune response, because it's foreign, it's bug cells inside of a bunny. The bunny think that this is not me, so it mounts an immune response. So imagine that these green things here are now invading. They're part of the rabbit's body, but they're from the fruit fly. So the rabbit has this immune response against it. And then you can you actually just do a blood draw from the bunny, and you process that blood, and you can get antibodies. So now you have antibodies that bind to the cell or cell types or whatever that you injected into the bunny. Your hope is that it only binds to that one very specific thing that you injected. You guys with me so far? Jimmy, I'll get you in just a second, my friend. And I know there's some questions, but these are like running quickly in my head, and I will definitely get lost if I stop. So, Matt or Matthews, thank you so much for that follow. Welcome on in. How are y'all doing today? And oh my goodness, we got a raid. Chat, we got a raid. Raiders, welcome on in, Alan